Hi, my name's Alex and I'm a PhD student at the Australian National University in Canberra, Australia. My poster is about RISER, which is a software tool which allows the real-time in silico enrichment of RNA species from nanopore signals. So I'll just start off with a really high-level overview of how RISER works, and then I'll go into more details of the poster. So as you can see in the top left here, there are several different RNA species inside the cell. So in blue, we have messenger RNA, which encodes for proteins. And in orange, we've got a variety of non-coding RNA types, which have a variety of types and functions inside the cell. So when an RNA sequencing library is prepared, all of the species that have poly A tails, which includes both coding and non-coding RNA, get included in your sequencing sample. So if you're only interested in a single RNA species, this is a problem because the number of reads you're interested in will be diluted. And RISER closes this gap computationally. So the way that it works is as the molecule traverses the nanopore, RISER assesses the first few seconds of the signal that's produced. And in real time, it's able to classify whether the RNA is coding or non-coding, and then use that decision to tell the sequencer to either accept or reject the particular RNA strand. So in this way, RISER is able to effectively enrich or deplete the coding or non-coding RNA species. Now, if we take a bit of a closer look at the poster, we'll start off with the motivation. Standard nanopore sequencing libraries for direct RNA contain several different RNA species with 3' poly A tails. But if you're only interested in a single species, you don't want your library to be diluted with the RNA that you don't care about. So RISER overcomes the need to be using these biochemical-based methods with a purely computational approach to only sequence the species that you're interested in. So the technical basis for RISER lies in the Nanopore sequencer. So Nanopore sequences individual RNA molecules directly and allows sequencing decisions to be made per molecule directly from that electrical signal. Now, the biological motivation for the project and the basis for why RISER works is the fact that messenger RNA has a characteristic 3' end, and this can be exploited to distinguish coding from non-coding RNA. The important point here is that Nanopore always sequences RNA from the 3' to the 5' direction. So the first part of that raw electrical signal from the nanopore is always going to capture the same 3' characteristics that can be used to distinguish between coding from non-coding RNA. So we've used a convolutional neural network model which was trained with around 1 million signals from a variety of human cell lines. So we systematically optimized the neural network architecture and its hyperparameters with a held-out validation set and ended up with this network here. So for more details of how we developed this model and what other types of convolutional neural network architectures we tried, have a look at my long talk in the iRNA track. So the performance of the model. We evaluated it on an independent test set which hadn't been used for training or any hyperparameter tuning, so that we could evaluate the model's capacity to generalize to unseen data. And we achieved an accuracy of 88% and an area under the curve of 0.96. Having developed the model, the next step is to integrate it with the read-until API, which is provided by Oxford Nanopore, to enact the real-time sequencing decisions. So the way that it works, we have the minion on the left, which is the sequencing device. We have Minnow in the middle, which is the commercial software provided by Oxford Nanopore to control the sequencing device. And then we also have the read-until API, which is provided by Oxford Nanopore to expose some functionality in Minnow to allow third-party software to control the sequencer. So that's where RISER fits in. So in terms of the enrichment that RISER is capable of producing, we have tested this using RISER during a live sequencing run in real time on a MinION device. So we ran the run and also RISER for 24 hours, and then we mapped the sequenced reads to the transcriptome. We found that RISER is able to effectively reduce the length of off-target reads, and you can see this here. So when the target was coding, the length of the coding reads was significantly different to that of the length of the non-coding reads, and that coding reads were much longer than that of non-coding. And the same effect was observed when non-coding was the target. So what this tells us is that RISER is able to reject the reads that are off target, effectively reducing their read length so that the sequencer has more time to sequence the target species. We found that when the target was coding, RISER was able to achieve a small enrichment of coding and a large depletion of the non-coding relative to a control run where RISER was not run. When non-coding is the target, RISER achieves around a 2.6x enrichment of non-coding and a depletion of your coding reads. So thank you for listening to my poster talk, and if you'd like to get more information, please visit my 20-minute long talk in the RNA track about RISER. Thanks for listening and hope to see you soon.